Hello, this is HC. This is complex genetics, and this is when we actually see different types other than normal genetics. We've already sort of talked about dominance. Dominance, of course, is when we have one allele that masks over the outcome of the other. We're going to talk about incomplete dominance. We're going to talk about codominance. We're going to talk about multiple alleles, and then finally we're going to talk about sex-linked genetics. Dominance, just when one dominant allele masks over the recessive one. Heterozygous genotype still looks like the dominant genotype, but there may be like a carrier. Example, like a human eye color, brown eyes are dominant over blue. What's the genotype for a person with brown? It can be either big B or big B, little b, because the little b is masked over by the big B, brown-eyed. Complex genetics. Different types of inheritance that do not follow the simple rules of dominance. For complex genetics, it is important to remember that heterozygous genotype will show a new and different type of phenotype. So it won't be masked over completely, or it'll actually be changed into a different kind. Example of this would be like red cows, which are RR, white cows, WW. Both are going to be purebreds, both are going to be dominant purebreds. And then we have the roan cow. Here are examples of a red cow and a white cow, and if they were to mate, actually you wouldn't have two cows, but one of these was a bull, you'd end up with this red-white cow. So both the red color and the white color show up, but it's a different phenotype than either of the parents. Incomplete dominance, when a dominant allele does not completely mask over a recessive allele. Heterozygous genotype will show a mixture or combination of the dominant recessive type. An example of this would be a flower color. In carnations, if you take a red flower and a white flower and you mate them, you get the red and the white. So you get pink carnations. Examples: red, white, and pink. Human example would be hair texture. Curly hair is incompletely dominant to straight hair. And then write the genotypes for curly, wavy, and straight. Curly would be either this kind, actually it is this kind, wavy, a little bit of both, and then the straight hair would be lack of the curly gene. And if you take a look at it, straight hair is usually round and widest. Wavy hair is oval shaped. Curly hair is even more oval, and a spiral curled or coiled hair would be even more oval. Follicle size for thick hair, thin hair, thick curly hair, and thin curly hair. So if two wavy people had babies, what type hair would their children have? So wavy hair is big C, little c for mom and dad. Put them together, we get a curly child, 25% chance of a curly child. We have a 50% chance of a wavy child, and we have a 25% chance of a straight-haired child. And again, this is when we actually have these kind of setups, you have a 25% chance each time there's a baby for it to be curly, or a 50% chance. Why more of the kids are actually going to be wavy haired if you did have two homozygous, or excuse me, heterozygous parents. Curly, wavy, straight, how does it look? Here are three examples. This guy's name is Juan Epstein. It's not his real name, but he was in a show called Welcome Back Carter back in the 80s, probably, maybe even late 70s and 80s. He's passed away by now. He actually had a heart attack, I think about 10 years ago now. This is Jessica Biel with wavy hair, and I'm not sure who this blonde is, but she's got very straight, very thin hair. But those are the examples of incomplete dominance of human hair. Codominant. Codominance when both alleles are equally expressed. That's the roan cow. So you actually do get both cows showing up as actually being white or red, but they're not going to be pink cows. Heterozygous genotypes show both the phenotypes at the same time. Cattle example: red hair is dominant with white hair, or it's codominant with white hair, and the roan term is actually when you should have both red and white hairs in the cow. And across roan cow with a white bull, and you get even more white, which is really weird. Sickle cell anemia is a codominance type in humans. Sickle cell is when the normal protein, and we did this with DNA, it's when the normal protein actually changes, and it changes the shape of the blood cell, and it can lead to lots of problems, especially if you take a look, the blood moves through these nice small capillaries. Sickle cell tends to uh, cause a roadblock, and that actually causes the blood to stop, could actually kill the downstream or cause lots of pain. Usually what it means is a blood transfusion where you pull out the sickle cell blood and you put in healthy blood, but sooner or later body is going to replace the dead donated cells with sickle cell, because that's the protein they produce. Sickle cell is inherited by codominance. Heterozygous ones, sickle cells are the carriers. They do not suffer from the symptoms of sickle cell anemia, but they do have the genetics for it, and they can pass it on to their kids. So what genotype would carriers have? Normal blood, but have the sickle cell trait. They would be big S, little s. Big S is normal, and little s is actually having the sickle cell. So let's cross two carriers. So we have a male and a female, and this one is not a sex-linked one. It can actually be male and female in the same ratio. So what we do is take mother's genes. Mother has a big S, little s. Dad has a big S, little s. 25% big S, normal blood. 25% big S, little s, normal blood. 50% big S, little s, little s. Big S, little s, and we have 25% for little s for sickle cell. So 25% normal blood, 50% normal blood except you're a carrier, and 25% sickle cell. Multiple alleles, these are when two alleles, more than two alleles actually go out for the trait. A person can carry only two of these alleles, and an example, this one is human blood type. Blood types actually come in type A, where you either have a big A or big A, big O, and let's just say AA or AO. B type, where you have BB, BO. O type, it's going to say C. The only type you can have that is when you have O on both sides, because if you have one of the other alleles, you have A or B. And then you can have AB, which again is only A and B, and we always write AB, not BA. So a woman who's heterozygous for A, that means A and O, marries a male who's O blood, two O's. So we give AO and we give O's. And A blood looks like this, and A blood has antibodies on the outside of it, and this allows it to only like A blood. And then we have O blood, and you can see this O blood antibody is different. If you gave this one to this one, it would actually look okay, but if you gave this one to this one, it wouldn't, and you'd have a problem. So O can donate to A, but A can't donate to O. And then we have this one over here, which is the same, and then we do this one, we have O blood again. So what percentage of their children will have type O? Looks like 50%. Along with the A's and the B's and the O's, we actually have negatives and positives. This is the RH factor. And we can actually have A positive and we can have A negative. And you can see the A positive actually has antibodies on the outside, and the negative has antibodies on the outside. And then we can do the same thing with B. And what makes it look different are these, instead of the squares, you have these points. And then a negative only has points, just like the A positive only has the squares. O blood has these concave things, which you can see in there and there. And then A negative has no antibodies whatsoever, which makes it very, very, very nice kind of blood to have if you're anything else. Not so good if you need something, because anytime you have these antibodies, you can't donate to O. And then AB blood actually has the A's and the B's, and the B negative blood has the A's and the B's without the C, or out the O, or without the positive, I guess. That's a positive one because O doesn't have it. So this makes blood positive. And which one makes it negative? Looks like either one of those. And if you look at the percentages in blood, O positive, this one makes up most of the blood, almost 40%. I am A positive, so is my wife and both kids, so we make up a pretty good share. And you can see some of these drops are actually really small, especially this AB negative. AB positive, not much. Negative blood actually seems to be less than the positive blood. Not sure why. Probably why we call the positive positive though, because that's probably the first one we found. This is the donor chart, and if you take a look at this donor chart, the way it works is this is the recipient side. So if you're an A plus, you can receive blood from O negative, O positive, A negative, and A positive. If you're AB, you can accept from anyone, and if you're O, you may donate to anyone. O negative, donate to anyone. Remember, the O negative has no antibodies. Negative blood can only accept from other negatives, and if you take a look at the negative, looks like negative, 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 and negative, negative, and it gets less and less as we go down. O negative can only get blood from O negative, which is really bad. O negative makes up 6.6% of the population, so they're in need of O negative blood a whole lot. So if you do have O negative blood, you can probably donate it, and it would be well worth it to someone. Letters can only donate to like letters, so A can only donate. If you take a look at A, how does this work? A, O. If you actually look at A's, letters can only donate to like letters. A positive can accept blood from anyone. O negative can donate to anyone. So it's really nice to be on this one if you're donating. It's really nice to do this one if you're accepting. Sex-linked traits. These are a little bit different because these are only on the 23rd pair. They're only on the X and Y chromosome. In fact, they're really only on the X chromosome because the Y chromosome really doesn't have anything that can go wrong. It just doesn't have very much material on it. To write the genotypes for sex-linked traits, you must include the sex chromosomes. Sex chromosomes are the gender ones. These are the 23rd pair. The other 22 are what are called autosomal, and we'll do that when we do 8D, when we start talking about pedigrees. There they are. This is the X chromosome. This is the Y chromosome. Looks almost like a Y, looks almost like an X, like all the other 22 pair. And we write males X and Y, and we write females as X and X. Mom gives the child an X, and whatever the dad gives, either a Y or an X to the child, it becomes either male or female. Men give their Ys to their sons, and they give X's to their daughters. So let's talk about color blindness. Color blindness is a recessive trait. It's found on an X chromosome. The X from the 23rd pair, so it comes from mom. What are the possible phenotypes and genotypes of color blindness for both males and females? Actually, it can come from the dad if the dad's for daughter. So, for females and for males, the genotypes, to make a normal female, we have an XX, and the phenotype there is normal vision. To make a normal male, we have an X and a Y, and we have normal vision. We have a color blindness X and a regular X, which this is a recessive trait, so the normal side is dominant. So we're going to have this lady would be a carrier, normal vision. And if we do this one, we have an X chromosome that's faulty, and we don't have another one. This guy is going to be colorblind. And if we do this one, we get a bad X from mom and dad, and this female will be colorblind. We won't do this one again because it's the same setup for there. This is an example of red and green color blindness, where you can't really make the difference between oranges and reds and greens and kind of turquoises. So as long as you can actually make out numbers for all these, you are probably not colorblind. If you are colorblind, you can't see the green, then everything looks the same. And if you can't see red and green, everybody looks the same in there too. Sex-linked traits, last one we're going to talk about. What if we have a woman carrier Color blindness, she marries a man that is colorblind, so mom's a carrier, she's got a bad X, but she's got a good one, so she can see fine. The man is colorblind, so has the bad X. So we put the bad X up there for the female, or for the male, and we put the bad X in there for a female. And what do we get? This one, because it's got a good X, she can see fine. Go down to this one because we have a bad X from dad and a bad X from mom. This girl is colorblind. Go down here, we have a normal vision male, and then go down here because there's only one X in the picture. So what percentage of the sons will be colorblind? 25. What percent of the daughters will be colorblind? It also is 25% chance. So even if you are a colorblind male and a colorblind carrier for female, you still have a 25% chance of a normal and a 25% normal male and female. So chances are you're still going to have a 50-50 chance of having a healthy visioned son or daughter as opposed to a non-healthy. Sorry, we have one more, hemophilia. Hemophilia is when normal blood has red blood cells and white blood cells and plasma, as well as something called platelets. And then when we actually break the vessel, these go out and in contact with oxygen or air, they start growing this activated platelet clot material. It's fibrin, it's a protein, and it ties up all these red blood cells and makes a clot, which we call scab. People who have extreme cases can bleed a lot. They don't have the ability to make the clots of the scabs, and it is a sex-linked disorder. It is on the X chromosome, and it can be passed down, but again, more or less, it only affects the male because the female can actually have another chance of getting it. Hemophilia, there is no cure. They have to avoid injury. They have to take medication. If they actually do bleed, they can get clotting factors. I've actually donated platelets many times, which they do give to patients who have blood disorders. Good nutrition is really good for them. Good dental hygiene, so we don't get bleeding gums because that could be a problem. We have intracranial hemorrhaging. If they get hit in the head, prolonged nosebleeds, they have bruises, they have painful swelling in their joints, and they have GI hemorrhaging. What these three things are, vomit, throwing up, they actually have coffee ground flavored or scented vomit. Their urine is cola colored, so it's really dark, probably because of blood, and very tarry stools. Normal man whose father was who had the disease, hermaphrodite, God, I hate this, marries a woman who's a carrier. What's the probability the sons will be hemophiliacs? And then what about their daughters? We have looks like a normal male up here. We have a carrier female. We can have normal. We can have normal, but a carrier. We have normal, and then we have a 25% chance of having a hemophiliac son. So what's the probability of their sons? 50%. What's the probability of their daughters? 0. But you do have to remember that this daughter right here will be a carrier. And here it would look like if you had the picture. Here's an unaffected father. There's the carrier mother. And we have an unaffected son, 25% unaffected daughter, 25% unaffected daughter, except it's a carrier for 25%, and then we have an affected son, 25% chance. So this is a really bad disease, and again, we'll talk about this in the bloodline when we start talking about AD and pedigrees. Okay, that's it. Thank you. Bye.